Welcome back to Weekly Neurology Deep Dive. This week, we're diving into emerging mechanisms and therapeutics in inflammatory muscle diseases. It's from Trends in Pharmacological Sciences. Just as a quick reminder, this deep dive is purely for educational purposes. Doesn't constitute financial or medical advice. We're really just summarizing some key points from the article here. And we strongly encourage you to read the full paper after listening to this. This deep dive is really geared toward giving you, as practicing neurologists, the most relevant information for your clinical practice.
Inflammatory myopathies or myositis can be incredibly challenging to treat, and that's because we need a deeper understanding of the disease mechanisms to develop truly effective therapies. This is especially true for inclusion body myositis, or IBM, which has been notoriously resistant to our current treatments.
The authors highlight how recent research advances like transcriptomics and a deeper understanding of antibody-mediated immunity and interferon signaling are paving the way for new therapies. What are some of the most promising therapies being explored right now?
We're seeing a lot of interest in chimeric antigen receptor T cells, or CIR T cells, and Janus kinase inhibitors, also known as JAK inhibitors. It's exciting because these therapies target specific immune pathways, potentially leading to more precise interventions with fewer side effects.
That shift towards targeted therapies is a real game changer. The article dives into different myositis subtypes,
Yeah.
but it seems to really focus on IBM. Why is that?
IBM is unique because it presents with both immune system dysfunction and muscle degeneration. This dual nature makes it incredibly complex to treat. Plus, it progresses slowly and often leads to significant disability. So the need for better treatment options is urgent.
Speaking of immune dysfunction, the article points out that T cells are the predominant immune cell type, found in muscle biopsies across various myositis subtypes. This is especially prominent in IBM, where cytotoxic T cells, also known as CTLs, are thought to be the main drivers of inflammation. What's interesting is this finding that a significant portion of these CTLs in IBM show signs of aging.
The researchers found that these aging CTLs express high levels of KLRG1, a marker typically found on older, less active T cells. This raises the possibility of targeting KLRG1 as a way to specifically eliminate these potentially harmful T cells without disrupting the regulatory T cells that help keep the immune system in check.
That's an incredibly targeted approach. The article mentions a phase 2/3 trial already underway to test a monoclonal antibody targeting KLRG1 called ulvaprubart. We'll definitely be following the results of that trial closely.
Absolutely. It's just one example of how a deeper understanding of the immune mechanisms in myositis is leading to novel therapeutic approaches.
Moving on from T cells, the article delves into the role of interferon signaling in myositis. It states that both type 1 and type 2 interferon signaling pathways are activated in myositis, leading to a surge in research on therapies aiming to reduce interferon activity. What are some of the potential therapeutic targets they discussed?
One promising avenue is JAK inhibitors. These drugs modulate both type 1 and type 2 interferon signaling and are showing potential in treating dermatomyositis and antisynthetase syndrome. For example, tofacitinib, a JAK1 inhibitor, has shown positive results in dermatomyositis patients, especially those with anti-MDA5 antibodies.
The paper mentions that ruxolitinib, a JAK12 inhibitor, is also being studied in a phase 23 trial for IBM. Are they focusing on any other approaches to targeting interferon in myositis?
Another exciting option is anifrolumab, an antibody that specifically targets type 1 interferon receptors. It's currently in phase 3 trials for both dermatomyositis and polymyositis. The diversity of approaches really speaks to the level of research being poured into targeting interferon signaling in myositis.
It's encouraging to see such a robust pipeline of potential therapies.
Right.
Now, the article shifts gears to discuss B cells and antibodies, which are also key players in the immune response. It emphasizes how myositis-specific antibodies, or MSAs, have revolutionized our understanding of myositis. How have MSAs changed the way we approach this disease?
MSAs are incredibly valuable tools both for classifying myositis subtypes and for guiding treatment decisions. For example, the presence of specific MSAs can help us predict which patients are more likely to respond to certain therapies.
The article talks about how, while we don't have a complete understanding of how MSAs actually cause myositis, research points to some possible mechanisms.
In dermatomyositis, for example, antibodies might be increasing type 1 interferon expression. Specifically, antibodies against MDA5 could potentially activate MDA5, leading to an increase in type 1 interferon. In immune-mediated necrotizing myopathy, antibodies against HMGCR and SRP may be directly damaging muscle fibers and increasing inflammation.
It looks like the specific mechanisms can vary depending on the myositis subtype and the specific MSAs involved.
Exactly. And in antisynthetase syndrome, we see antibodies targeting aminoacyl tRNA synthetases, which are essential enzymes for protein synthesis. Depending on the antisynthetase syndrome subtype, these antibodies could be contributing to the disease in a variety of ways.
Now, IBM presents a slightly different picture when it comes to antibodies. The role of antibodies, particularly those targeting CN1A, is still debated in IBM.
That's right. While anti-CN1A antibodies haven't proven to be very sensitive or specific for IBM, mouse models using CN1A immunization have been able to replicate some of the hallmark features of the disease. So the jury is still out on the exact role of anti-CN1A antibodies in IBM.
Despite the uncertainty surrounding their role, researchers are still actively exploring ways to target B cells and antibodies in myositis. The article highlights a few different approaches.
One approach that's been gaining traction is reducing antibody levels by targeting B cells and/or plasma cells, especially in myositis subtypes with specific MSAs.
Rituximab, which targets CD20 on B cells, is already commonly used to deplete B cells in various autoimmune diseases. However, the article points out that it faces limitations when it comes to tissue penetration, which could hinder its effectiveness in some myositis subtypes.
That's where anti-CD19 CRT cells come in. They show much better tissue penetration compared to rituximab, and have already demonstrated impressive results in severe treatment-resistant antisynthetase syndrome cases. Trials are underway to evaluate their efficacy in other myositis subtypes as well.
That's exciting to hear. Are there any other B cell targeted therapies being explored?
Daratumumab, a monoclonal antibody targeting CD38, is another interesting option. It hits both B cells and plasma cells and has been successful in treating severe dermatomyositis and immune-mediated necrotizing myopathy. The article even suggests it could potentially be a treatment option for IBM patients with anti-CN1A antibodies.
So we're seeing a lot of progress in developing B cell targeted therapies. What about other strategies that focus on different aspects of the immune response?
Researchers are also investigating approaches like targeting BAFF, a cytokine involved in B cell development. Trials with belimumab, a BAF neutralizing antibody, haven't shown success in myositis yet, but it's still an area of active research.
Are there any other potential targets on the horizon?
BTK inhibition is showing promise in other autoimmune diseases, and preclinical data suggest it could be beneficial for antisynthetase syndrome, or anti-MDA5 dermatomyositis with lung involvement. Another strategy being explored is Ig-lowering therapies, such as efgartigimod alpha. This approach has shown positive results in other diseases and immune-mediated necrotizing myopathy models, leading to trials in various myositis subtypes.
It's remarkable how many different avenues researchers are pursuing to target the immune system in myositis. It really highlights the complexity of these diseases and the need for a multifaceted approach to treatment.
Now, shifting back to IBM, we've touched on the debate surrounding whether it's primarily driven by inflammation or degeneration. The article delves deeper into this question, highlighting how protein aggregation is a key feature of IBM.
They specifically point to TDP43 and P62, two proteins that form aggregates in IBM. We also see these aggregates in other neurodegenerative diseases like ALS and frontotemporal dementia.
And interestingly, the article mentions a study where reducing T cells in a mouse model of IBM didn't significantly affect myofiber degeneration. This finding further supports the idea that there's a degenerative process happening in IBM that's at least partially independent of the immune response.
But the immune system might still be playing a role in IBM, even if it's not the primary driver of muscle damage. The article highlights a study using single-nucleus RNA sequencing, or snRNA-seq, along with spatial transcriptomics to investigate IBM. The study revealed that type 2A muscle fibers are particularly vulnerable in IBM compared to other myositis subtypes.
The researchers found that damage to these type 2A fibers was associated with increased expression of GADD45A, a protein involved in stress responses. And even more intriguing, this GAD45A expression correlated with T-cell infiltration.
So even though simply depleting T cells didn't prevent myofiber degeneration in the mouse model, it's possible that T cells are releasing signals that trigger stress responses in these vulnerable muscle fibers, ultimately contributing to their degeneration.
It's a complex interplay between inflammation and degeneration, and this study provides valuable insights into how these two processes might be interacting in IBM.
The article goes on to discuss another puzzling aspect of IBM denervation. This is frequently observed in electromyography studies of IBM patients. but its relationship to the disease process has been unclear.
A recent study shed some light on this issue. Researchers found increased expression of acetylcholinesterase, or ATE, in the muscle fibers of IBM patients. What's intriguing is that this ACE's expression wasn't localized to the neuromuscular junctions, where you'd typically expect to find it.
And what's the significance of this finding? Could it be related to the denervation observed in IBM?
The article suggests that this increased ATE activity occurring outside of the neuromuscular junctions might be disrupting neuromuscular signaling, ultimately leading to denervation. They even propose a potential mechanism involving a long, non-coding RNA called NORAD.
For those who might not be familiar, long non-coding RNAs or LNC-RNAs are RNA molecules that don't code for proteins, but they play important roles in regulating gene expression. How does this NORAD LNC-RNA fit into the picture?
It seems that NORAD, which is often upregulated in response to genomic stress, can inhibit a group of proteins called proteins. These proteins, in turn, regulate the translation of various proteins, including ACE.
So if NORAD is inhibiting proteins, it could lead to increased translation and activity of AC. Is that how they're connecting it to the denervation seen in IBM?
Exactly. The idea is that genomic stress, which is common in many diseases, including IBM, leads to an increase in NORAD expression. This, in turn, inhibits proteins, leading to increased production and activity of ACE. And this excess ACE activity outside of the neuromuscular junctions could be interfering with normal neuromuscular signaling, resulting in denervation.
It's like a chain reaction, starting with genomic stress and ending with the innervation. That's a fascinating insight into the complex interplay of molecular mechanisms in IBM.
And there's another cell type that's gaining attention in IBM research, fibroadipogenic progenitor cells, or FAPs. These cells are involved in muscle regeneration, but in IBM, they can become senescent.
Senescence refers to a state where cells are no longer able to divide and function properly. How does this relate to IBM?
A study mentioned in the article found that these IBM-specific senescent FAPs upregulated P21, a protein that promotes cell cycle arrest and senescence, as well as pro-inflammatory genes like C3.
So not only are these senescent FAPs not contributing to muscle regeneration, but they're also pumping out inflammatory signals, potentially exacerbating the inflammatory environment in IBM.
Precisely. This makes senescent FAPs a very interesting therapeutic target. If we can find ways to prevent them from becoming senescent, or to eliminate them altogether, we might be able to slow down the progression of IBM.
It's like tackling the degenerative and inflammatory aspects of IBM with one stone. Now, shifting gears a bit, the article also discusses some really exciting advances in transcriptomics and how they're revolutionizing myositis research.
Transcriptomics, which involves studying all the RNA transcripts within a cell or tissue, is giving us a much deeper understanding of the molecular underpinnings of myositis. It's like getting a snapshot of what's happening at the gene expression level, providing insights into which genes are turned on or off in different cell types and disease states.
They mentioned a study that used bulk RNA sequencing followed by machine learning to identify transcripts that could accurately classify muscle biopsies into different myositis subtypes.
The accuracy was remarkable. For example, they could classify muscle biopsies from patients with antisynthetase syndrome, dermatomyositis, and immune-mediated necrotizing myopathy with an accuracy of at least 91%, just based on their unique gene expression pattern.
And that level of accuracy is truly impressive. It suggests we're moving towards a future where we can diagnose myositis subtypes with a high degree of certainty using gene expression analysis.
And that's just the tip of the iceberg. The article also delves into the power of single-cell RNA sequencing, or cRNA-seq, which allows us to analyze the gene expression profiles of individual cells.
That's incredible. It allows us to see how different cell types are contributing to the disease process, providing a much more granular understanding of what's happening at the cellular level.
One study using scRNA-seq analyzed peripheral blood mononuclear cells, or PBMCs, from individuals with antisynthetase syndrome and dermatomyositis. They identified specific cell populations with unique gene expression signatures, suggesting these cells might be playing key roles in the disease process.
That's really insightful. It seems cRNA-seq is not only helping us to identify potential therapeutic targets, but also to uncover biomarkers for disease diagnosis and prognosis.
Absolutely. One of the most intriguing findings from this study was the identification of ISG of 15 plus cytotoxic T cells as a potential prognostic biomarker for anti-MDA5 plus dermatomyositis. This means that the presence of these specific T cells could help us predict how patients with this subtype of dermatomyositis will fare.
It's remarkable how transcriptomics, especially when combined with single-cell analysis and machine learning, is transforming how we approach myositis research.
It's allowing us to unravel the complexities of these diseases at an unprecedented level of detail.
Welcome back to Weekly Urology Deep Dive. We're here for the final part of our deep dive into emerging mechanisms and therapeutics in inflammatory muscle diseases. Over the past 2 weeks, we've explored the latest research on inflammatory myopathies, from the roles of T cells, interferons, B cells, and antibodies, to the unique challenges of IBM and the transformative power of transcriptomics.
As we wrap up this deep dive, it's helpful to zoom out and consider the bigger picture. What are the key takeaways from this review?
One of the most striking themes is the sheer complexity of these diseases. Myositis isn't a single entity, but rather a spectrum of disorders with overlapping yet distinct mechanisms. This emphasizes the need for personalized medicine, tailoring treatment to the specific subtype, and even the individual patient.
It's a move away from a one-size-fits-all approach towards a more nuanced understanding of how to effectively manage myositis. The research highlighted in this article demonstrates the incredible progress made in identifying key pathways involved in myositis. This has paved the way for the development of targeted therapies like JK inhibitors, which are showing promise in treating dermatomyositis and even IBM.
We've seen a similar shift towards targeted therapies in the realm of B-cell targeted treatments, with anti-CD19 CAR T cells and daratumumab offering hope for patients with severe treatment-resistant myositis. It's encouraging to see these advances, particularly in IBM, where traditional immunosuppressants haven't been very effective.
The focus on targeting both inflammation and degeneration in IBM is indeed leading to novel therapeutic approaches. Sirolimus, for instance, which targets both of these processes, is showing promise in improving patient outcomes. The research on IBM also emphasizes the importance of understanding the interplay between inflammation and degeneration in this complex disease.
And as we discussed earlier, the role of non-immune cells, such as type 2A muscle fibers and FAPs, is gaining increasing attention. This opens up a whole new realm of therapeutic possibilities as researchers explore ways to protect these vulnerable cells from stress and senescence.
This leads us to another key takeaway: the transformative impact of transcriptomics on myositis research. We're now able to analyze gene expression with remarkable precision, allowing us to identify specific biomarkers for diagnosis and prognosis.
The study that used bulk RNA sequencing and machine learning to classify muscle biopsies into different myositis subtypes with over 90% accuracy is a prime example of how this technology is revolutionizing diagnosis. The emergence of single-cell RNA sequencing adds another layer of insight, allowing us to dissect the roles of individual cell types in the disease process.
The integration of transcriptomics with single-cell analysis and machine learning is truly ushering in a new era of personalized medicine in myositis. It's fueling the development of novel diagnostic tools, allowing for more precise subtyping and personalized treatment strategies, and it's helping to identify potential therapeutic targets that could lead to more effective treatments.
So as we conclude our deep dive into emerging mechanisms and therapeutics in inflammatory muscle diseases, it's clear that we're in the midst of an exciting and rapidly evolving era in myositis research. The pace of discovery is accelerating, and new treatment options are emerging at a rapid rate.
While challenges remain, particularly in finding effective treatments for IBM, the future of myositis research is bright. The insights gleaned from transcriptomics, combined with a deeper understanding of the complex interplay between immune and non-immune cells in these diseases, are paving the way for a new era of personalized medicine that offers hope for improved patient outcomes.
We hope this deep dive has provided you with valuable insights into the latest advancements in myositis research. Remember to check out the full article for a more in-depth look, and stay tuned as we continue to explore the ever-evolving world of neurology.